Alright guys, so this is going to be part 1 of the population, evolution, and changing allelic frequency video. This first part is going to just deal with population evolution, and then I'm going to stop the recording there, and then I'm going to make a whole new recording for the changing allelic frequency. But don't forget, you're taking these down on your one set of notes that I handed out in class today. If I sound a bit nasally, or I sniffle, or I sneeze, or anything along those lines, kind of sick, but it'll be fine. Just go with it. Don't forget to pause whenever you need to. So here we go. Population evolution. So we talked about natural selection, but we have not yet really defined evolution. And the basic definition that we always give you is that evolution is change over time. But at this level, we consider evolution to be a change in the inherited characteristics of biological populations over successive generations. So it's still change over time. Here's your change. Here's your time. But specifically, we're talking about the characteristics that are inherited. So the genes that are passed on and the phenotypes that those genes lead to. It's going to depend heavily on what that environment selects for. Okay, what makes you more successful or makes you more fit. So there are two kinds of evolution. There is microevolution and macroevolution. Microevolution is a change in gene frequency within a population over time. And that change can be due to one of 4 or maybe multiples of 4 different processes. Mutation, selection, and selection can be either natural or artificial, gene flow, and genetic drift. Now, you guys at this stage have already done your goldfish lab. So the same gene frequencies that we were calculating in that goldfish lab, where you have 2 gold ones out of a population of 10, so that makes your gene frequency 0.2. That's pretty much what they're talking about here. In terms of macroevolution, macroevolution is the occurrence of large-scale changes in gene frequencies in a population over a geologic time period. So it's kind of like microevolution, but it's taking an even longer time. So think of it as macroevolution has the ability to change gene frequencies in a population over a period of time, fairly small period of time actually, whereas macroevolution is happening over an extensive period of time. So we're talking hundreds of thousands of years and eons and epochs and stuff like that. So you already know that a population is a group of the same species who live in the same environment and are capable of mating with each other. So it's a localized group of interbreeding individuals. The gene pool is the collection of all the different alleles in a population. Now don't forget there is a difference between genes and alleles. Gene is a section of DNA that codes for something, and allele is the version of the gene you get. So for every individual, they're going to have 2 alleles of their genes. They get one of those alleles from mom, and one of those alleles comes from dad. Allele frequency is how common is that particular allele within the population. So for example, how many capital A's do we have in a population versus how many lowercase a's do we have in a population? Alright, so how do we determine allele frequency? So I want to draw your attention to this picture. We have a bunch of different frogs. Most of them are green. We have a couple of red and a few that are purple or blue, whatever color you want to call that. Their color is determined by one gene. One section of DNA that codes for skin color. That gene has 2 alleles, and those 2 alleles exhibit what we call incomplete dominance. Hopefully you remember this from your freshman days, but just to give you a refresher, with incomplete dominance, your heterozygote is a completely different color from mom or dad. So this version of the gene codes for green, whereas this version codes for red, and this one codes for blue. So at this stage, we're not really writing any of this down. We're just kind of discussing and talking about it. Okay, this is the stuff you write down. So how do we count alleles? Well, we have to make the assumption that there are going to be 2 alleles for the gene. a capital allele and a lowercase allele, also known as the dominant allele and the recessive allele. The frequency, how often we're seeing that dominant allele, is given the variable p, lowercase p, whereas the frequency of the recessive allele is given the variable q. The frequencies must add up to 1 or 100%, so that's where we come up with the formula of p plus q equals 1. So if we look at the problem before, the one with the frogs, we had a dominant allele of big C, big G, and that would represent, or that would be represented by P, because it's the frequency of our dominant allele, whereas big C, big R would be the recessive allele, and that would be represented by the variable Q. The P and the Q come from the Hardy-Weinberg equation, which we haven't discussed yet, but we will at the second half of this video. So let's go through and look at this problem as it relates to P's and Q's and frequency and all of these. So these frogs are diploid, and because they're diploid, they have 2 copies of their genes for color. 1 copy came from mom and 1 copy came from dad. The total population is 16 frogs, but keep in mind that these frogs are diploid, which means that they have 2 copies of their genes for the allele, 2 copies of their genes for color, So we have 11 green frogs, which mean that if they are completely green, they are going to have 2 copies of the big C, big G gene, because that's the only way they could be green. They have to be homozygous dominant. So since there are 11 of them, technically there are 22 copies of this particular allele. Now, as far as the recessive allele, there'll be 0 copies of that because these green frogs are homozygous dominant, 2 of the dominant alleles. There are 2 blue frogs, and they have an even split of their alleles. They have 2 copies of the dominant allele, but they also have 2 copies of the recessive allele. These would be our heterozygote frogs, and they're blue. Our red frogs are our recessive, Our red frogs express our recessive trait, which means that they will have 0 copies of the dominant allele, but they'll have 6 copies of the recessive allele because there are 3 of them and they each have 2. So as far as the dominant allele goes, we have 24 copies of that particular allele. And we have 8 copies of the recessive. So to work out our frequency, it would be how many of those particular alleles we have divided by the total population, how many we could potentially have. So because we have 16 frogs, and each frog would have 2 alleles, we could have up to 32 alleles in terms of actual number. But the frequency for the dominant allele would be 24 over 32, which is 0.75%, or 75%, or 0.75, and the frequency for the recessive allele would be 8 over 32, which is 0.25 or 25%. So if these frequencies change, this is what the population is now, as it's represented by this picture. If these frequencies go up or down, if they change at all, it means that evolution is occurring. Change is taking place within this population. So populations do evolve, and we know that the mechanism by which this happens is natural selection. Now I want to make it abundantly clear that natural selection acts on individuals, not on the population. Natural selection acts on individuals, whereas evolution does not. So think of natural selection as differential survival, or survival of the fittest, meaning the more successful or the more favorable traits that you have that allow you to survive in that environment, the more likely you are to survive. There is also what we call differential reproductive success. So the individuals that bear more offspring pass on their traits that allow them to be successful, and those traits stay within the gene pool. Now, populations do evolve because the genetic makeup of a population can change at any point in time. Technically, that's evolution. what we tend to see is the more favorable traits, the ones that allow the individuals to survive for a longer period of time, become more and more common as the population evolves. These are just some examples of how populations can change over time. I like this one best because these guys are cute. But these are pocket mice, and it just shows how coloration will change depending on the environment. So a lava flow takes place. Excuse me, sorry guys. And as the lava cools and hardens, it becomes dark. Well, pocket mice, which are normally a sandy color because their environment is a sandy color, they will turn a darker, almost brownish black color so that they can blend in with their background. Okay, so again, this slide is just to emphasize the fact that individuals are selected. Either an individual survives or an individual does not survive. Either an individual reproduces or it does not reproduce. That's pretty much it when it comes to individual organisms within a species. They do not evolve. The individuals themselves do not evolve. Okay, so we talked about fitness in our last video, but we're going to mention it here again. Fitness can refer to two things. We can look at survival fitness, and we can also look at reproductive fitness or reproductive success. That's all that it boils down to. How successful are you? Did you live long, and could you reproduce? So both your survival and reproductive success can be determined by your phenotype. Individuals with one phenotype leave more surviving offspring than individuals that have multiple. And that's not really humans. We don't really fall into this category. But nearly everything else, that seems to be the going theme. So here we have a couple of graphs that all relate to this insect called a water strider. And we're looking at the female of the species. So we can see that as the female gets bigger, she lays more and more eggs per day. So that would be good. Bigger females lay bigger eggs. That sounds good. But if we come over to this graph, hold on, I need to adjust this just a little bit for me. Okay, if we come over to this graph, we see that the bigger the female, the less her lifespan is. So as the female gets bigger, her life expectancy or her lifespan in terms of how many days she is alive for decreases. So whereas bigger females lay more eggs, bigger females also die faster. Then if we go over here and look at this graph, we will see that bigger females, in terms of the length of the female, initially, the smaller they are, the more eggs they lay over a lifetime. But after they pass like 14, right here I would call this a critical point. After they pass 14 and they continue to increase in size, they actually lay less eggs during their lifetime. So this just goes to show that you kind of have to choose. Either you're going to be a big female and lay lots of eggs per day, but for a small amount of time and live for a very short amount of time, or you're going to be a smaller female, not lay as many eggs, but you live longer and you ultimately lay more eggs over your lifetime. So we know, sorry, the slide shifted. Hold on a second. Okay, so we know that natural selection takes place, but what's the driving force behind it? Well, variation within a population is the raw material or the driving force for natural selection. There has to be differences within the population so that natural selection can take place. Because of those differences, some individuals will be more fit than others. Some individuals must be more fit than others for natural selection to lead and prod evolution. So where does this variation come from? Well, it can happen very naturally in the form of a mutation. And all that a mutation is, it's a random change to your DNA. It can happen because of errors in mitosis and meiosis when the cells divide, or it can happen because of environmental damage, things like UV light and free radicals and stuff like that. Variation also comes from sexual reproduction, the mixing of alleles or the recombination of alleles. Every time sexual reproduction takes place, you end up with new arrangements in every offspring, which is why even though you and your brothers and sisters may both belong to the same mom and dad, you are not carbon copies of each other, nor are you carbon copies of your mother or your father. Each new combination results in a new phenotype. Now, is there some continuity? Yes, there is, because there are certain things that you will inherit from your mom. So variation spreads, but some things remain the same. Offspring do inherit certain traits from their parents. So now we're going to explore the 5 agents of evolutionary change. They are mutation, gene flow, non-random mating, genetic drift, and selection. Step through these one at a time. So starting off with mutation and variation. Mutations create variation, especially because new mutations are constantly happening. They're happening all the time. A mutation causes a change to your DNA sequence. If your DNA sequence changes, that can cause changes to your amino acid sequences and thereby your proteins, which will cause changes to structure and function, and ultimately will end up with a change in phenotype and therefore a change in fitness. Sometimes it's a good change, sometimes it's a bad change, but it happens regardless. Okay, gene flow. Gene flow is the movement of individuals and alleles in and out of populations. So think of seeds and pollen being distributed by wind and insects and animals and stuff like that. Think of the migration of animals from one part of an area to a completely different area. Different subpopulations may have different allele frequencies just depending on how many individuals are coming in versus how many are leaving. They're also going to have different gene alleles genetic alleles, because there's going to be genetic mixing as you go from one region to another. You're going to end up with reduced differences between populations if this happens continuously. We're seeing this a lot in America. The more people that are coming in and the more interbreeding, for lack of a better term, that takes place, the more and more we have more like a blended person, if that makes sense. My family is a perfect example of that, where we have all these different kinds of individual people, but more and more the offspring are looking kind of like me and my cousins. We're blended, even though my dad's half white and my mom's Portuguese and Indian and a bunch of other things. So, yeah. All right, so here's an example of what I was talking about. Gene flow is especially common in the human population. It's on the increase in terms of our human population. We're always transferring alleles between populations. So when I came from Grenada, I brought my genes with me, and now my genes are a part of the gene pool in America, in Texas, or in Mesquite, if you want to get specific. Okay, non-random mating. This is also known as sexual selection. It suggests that mating between males and females is not a random process. It's not like whoever next walks through that door, that's who I'm going to mate with. No. Females are looking for very particular or certain visual clues that showcase the vitality of their potential mate. Males that lack these characteristics don't really get to mate. So if you think of a lion, one of the characteristics would be a very thick and full mane, and normally one that has some kind of color variation to it. They particularly are attracted to like the black and the darker brown colors within, the lighter tawny color. This is an example of the frigate bird, which I think I mentioned once before. And these guys have a throat pouch, this red section here, that they can deflate. Normally it's deflated, and then when they're trying to attract a mate, they inhale and they expand that throat pouch using air, and the more vibrant red color, and the bigger the throat pouch, the more attractive it is to our female. Here's another example of, these are deer. Only the male grow antlers, and the wider and more impressive and bigger and heavier those antlers are, the more it speaks to him being a viable mate for that particular female. This one I've always thought of it as interesting. These are, of course, you probably recognize this as a peacock, and only the male of the species is as brightly colored, but the bigger the tail, the fancier the tail, the more vibrant the colors, the more it tells his potential mates that he is a good potential father for their offspring. Okay, genetic drift. So genetic drift has a lot to do with chanced events. Sometimes we think of it as the effect of chanced events, and there are two of them. The first one is called founder effect, and the second one is called bottleneck. So in founder effect, you have a small group of individuals that kind of break away from the main colony and move off and start their own little colony. Like Darwin's finches, for example. All of those finches were once just a regular, normal South American finch. And somehow they made their way over to the Galapagos Islands. No other finches followed except for them, and they diversified the area. The other one is called bottleneck. So with the bottleneck effect, some kind of factor, normally a disaster, reduces the population to a very small number. And then over time, that population recovers and it grows and expands once more. Right, so let's delve into these two a little deeper. Okay, so again, founder effect is when a new population is started, but it's only done so by a few individuals. When this happens, alleles that at one point might have been rare alleles, ones that didn't have a lot of frequency in the old population or in the old colony, for lack of a better term, may now be at higher frequencies, whereas others that were once prominent may now be missing. So what founder effect does is it skews the gene pool of the new population. A good example of this would be human populations that started from small groups of colonists. So for example, when we colonized the New World. Not when I say we, you know what I mean. Or like the Amish population. That would be another good example of founder effect. So this map shows the distribution of specifically the O blood type or the O-type blood allele. And it shows you where the original populations existed that had or showcased that particular allele. So the higher the area where it's found in the largest concentration is colored in blue. I want you to take note that it's mostly, or all actually, of South America, parts of the Caribbean, looks like Florida and some of the southern states, but not really Texas so much, and then Central America, all up through California. So that's where, and then of course there's a little bit in like the Alaskan area and out into the northern part of Canada. This is where the O blood type originated. Well, now that we know that there's O blood everywhere in the world, but it just goes to show you the migration route of these people. They started in this part of the world and they've moved until O blood is kind of found everywhere. Here's another one. This one shows the distribution of the B-type blood allele. So native populations look like upper parts of, like Russia, upper Europe, that kind of area. And then we have kind of down in the middle, close to like India, and we have some small little tiny subpopulations. But the B blood type, very concentrated, pretty much, Europe and Asia, and that's about it. But now we know that it's everywhere. What I find interesting is there's no trace whatsoever of the B blood type in Australia or even over in the Americas. And now, of course, we know that's not true. These people have also migrated and, B blood or B alleles are found kind of everywhere now. Still rare, but, still found all over the place. All right. Hopefully that you learned at some point in time that Africa is a cradle of human civilization. It's kind of where we originated. And this just kind of goes around and shows you all the different migratory paths that humans took out of Africa into the various parts of the world. Okay, bottleneck effect. So bottleneck effect is where you have a large population that is suddenly and drastically reduced by a disaster. So you go from having hundreds and thousands of individuals to having 50 individuals. And a lot of the times, this is due to some kind of natural disaster or natural occurrence, like a famine or a flood or an earthquake or a volcano erupts, or all of the habitat that that organism is found in gets removed or cut down or something like that. So this is a loss of variation by a chanced event. I mean, these animals didn't plan for this thing to happen. It just kind of did, and they happened to be in the wrong place at the wrong time. Because a lot of the individuals literally die, it means that alleles are lost from the gene pool. I want you to note one of the main differences between this and natural selection is this has nothing to do with fitness. Okay? For the most part, even if an animal was a fast runner, they would not be able to outrun a lava flow. So this has nothing to do with actual fitness, but this is just a complete, like a freak accident. Let's think of it that way. So what it does is it narrows the gene pool. So if you look at the visual, we started off with parents that had quite a bit of variation. And now, whatever event has taken place, and this is all of the variation that we have in the surviving members. So it means that when these repopulate, it's going to be pretty much carbon copies of each other. We're just going to have a couple of those alleles still existent, which means that variation isn't going to be huge. There's not going to be a lot of variation between the individuals of that new population. Right? A good example of this would be cheetahs. All living cheetahs as of now share a small number of alleles. It breaks down to them having less than 1% diversity between each other. So it's pretty much as if all the existing cheetahs are identical twins. They're all actually related because they're all interbred. And this happened because of two bottlenecks. About 10,000 years ago, when the Ice Age hit, a lot of cheetahs were affected. A small amount were left. The population increased based on just those individuals. And then within the last 100 years, this little thing called humans have hit the cheetahs. And now because of poaching and us encroaching on their habitat, they're right back at a bottleneck once more. These animals are so fragile from a variation perspective that any disease that affects one could ultimately affect all of them. We are within a hair's breadth of losing pretty much all of the world's cheetahs at this stage. And it's not because of a physical, like individual population thing, like numbers, but it's because of the fact that they don't have a whole lot of variation and they're all interrelated. Okay, so bottlenecking is an important concept when we think of conservation of biology, especially from endangered species. When we get down to having small, very small numbers of animals left, it means that we have to regrow populations from just a few individuals. So there is a loss of alleles from the gene pool. Some of those alleles were very successful ones, or ones that gave successful traits and phenotypes, but now they're completely gone. It reduces variation among the individuals, but it also reduces adaptability. There are only so many genes that these animals can pull from. They can only adapt so many different ways. Okay, natural selection. So we know that it's differential survival and reproduction, and it's due to changing environmental conditions. So things like changes in climate, the availability of food, predators and parasites and diseases, and toxins. Natural selection is governed by the combination of alleles that provide fitness. The more fit you are, the more likely you are to reproduce and thereby increase the population. All right, so those are the 5 agents of evolutionary change. Again, mutation, gene flow, non-random mating, genetic drift, and selection. Yeah, so hopefully there are no questions because I super can't answer them until you come to class next time. All right, so this is where part 1 stops. This would be a good time to go get a drink of water, maybe eat some dinner, play a video game for like 5.6 seconds. I'm just kidding. But, take a small break, relax yourselves a little bit, and then come back for part 2. Bye, guys. Okay, so this is where